Afternoon, students.
Good afternoon, sir.
So we'll continue today that the respiration. Okay, and here today we will say after that after the fermentation and glycolysis. Okay, today we will see what happened when there is oxygen in the system, and that is the when there is oxygen in the system, the product of the glycolysis, that is the pyruvic acid, that pyruvic acid is uh, completely oxidized. This pyruvic acid, this is completely oxidized and it forms ultimately the water and carbon dioxide with the naturally when it is completely oxidized, so there is formation of lot of energy. Okay, so there is you can see here the steps. Actually, that pyruvic acid, which is produced by the end of glycolysis, that pyruvic acid is not directly entered into the that Krebs cycle. Okay, this is this one is Krebs cycle, right? This Krebs cycle, this is not directly, but what happened? This pyruvic acid, it is converted into a compound called the acetyl coenzyme A. Acetyl coenzyme A, we will read it COA. Okay, we will read it as coenzyme A. So this pyruvic acid is converted into acetyl coenzyme A. And during this is not one step reaction, but this is a complex process, long reactions, and several enzymes, etc., take part in this. So it is called the pyruvate dehydrogenase complex. Okay, the enzyme is pyruvate dehydrogenase complex. And during this process, two things happen. One is a decarboxylation, and another thing is a dehydrogenation. Dehydrogenation. Okay. So by decarboxylation, there is a release of carbon dioxide, and by dehydrogenation, hydrogen is removed. This hydrogen which is removed, that is accepted by the coenzyme NAD, right? This NAD, it accepts the hydrogen, and this NAD forms NADH. Okay, so here you can see that NAD has been, this NAD has been consumed and NADH2 has been formed. Okay, in short, and here you can see that one molecule of carbon dioxide has come out. This is the first molecule of carbon dioxide that is generated in respiration. In respiration, we know oxygen is consumed and carbon dioxide is released. So this is the first step of the reaction where carbon dioxide has been released, the first molecule. And this one formed the pyruvic acid is converted into acetyl coenzyme A. This acetyl coenzyme A is the compound for Krebs cycle. Okay, so this is oxidized in the Krebs cycle. This is called Krebs cycle because Hans Krebs Hans Krebs, a biochemist who gave this detailed process of the oxidation of acetyl coenzyme A in a cyclic reaction, and so it is known as Krebs cycle. This is also known as the citric acid cycle. Citric acid cycle. Okay, because the first compound that is produced in the Krebs cycle is citric acid. All right, when this acetyl coenzyme A enter the Krebs cycle, and the first compound is citric acid. So it is called citric acid cycle. This is also known as TCA cycle. TCA cycle. TCA cycle is tricarboxylic acid cycle. Why it is tricarboxylic acid? Actually, these are the this several, but you can see here in the Krebs cycle there is a production of several acids. Organic acids are there. There is citric acid, isocitric acid, aconitic acid, oxaloacetic acid. Okay, all these are alpha ketogluteric acid, succinyl coenzyme A. Among these are acids with three carboxylic groups. Okay, so they are called the tricarboxylic acid cycle as there. So the Krebs cycle is known by, it is Krebs cycle, citric acid cycle, TCA cycle. All right, different name of this. So what happened here? This acetyl coenzyme A, it enter into the mitochondrial matrix. Okay, remember that Krebs cycle occurs in the matrix of mitochondria. Okay, in the matrix of mitochondria. So here is this one is mitochondria's involvement is there. The acetyl coenzyme A is accepted by oxaloacetic acid. This one is a 4-carbon compound. Okay, oxaloacetic acid. Remember, in the case of C4 cycle, also oxaloacetic acid is produced. This is 4-carbon compound, and this 4-carbon compound combined with acetyl coenzyme A, which is a 2-carbon compound, and they together form a six carbon compound citric acid cycle. This citric acid cycle undergoes several reactions and again generate the oxaloacetic acid. You can write OAA. Okay, oxaloacetic acid. This oxaloacetic acid combined with acetyl coenzyme A and ultimately forms the citric acid. The citric acid forms several intermediate compound and it undergo oxidation. So from the citric acid there is formation of oxalosuccinic acid. This is oxalosuccinic acid. During this process, a molecule of NADH2 is formed. Say here. So this is a step of oxidation where hydrogen is removed. From here, then oxalosuccinic acid is converted into alpha ketogluteric acid. Here, this is a decarboxylation reaction. Here, one molecule of carbon dioxide is released. So, this is the second carbon dioxide molecule released. And then alpha ketogluteric acid change into succinyl coenzyme A. During this process, another NADH2 is formed. So here also further alpha ketogluteric acid undergo oxidation. So NADH2 is produced. This one also undergoes decarboxylation. So carbon dioxide is also produced. So all the three carbon dioxide molecules are produced. One here during the conversion of acetyl coenzyme A. Second one is during the formation of alpha ketogluteric acid, and third one during formation of succinyl coenzyme A. So this from each molecule of pyruvic acid, three carbon dioxide molecules are produced, and here also one NADH2. This succinyl coenzyme A is converted into succinic acid. When it converts into succinic acid, there is a formation of GTP. This GTP then change into ATP. From the GTP, ATP is synthesized. So we can take this step as a formation of ATP here directly at the substrate level. Succinic acid again undergo dehydrogenation and form one reduced coenzyme FADH2. Okay, here not NADH2, here it is FADH2. And then fumaric acid through two uh, reaction, it forms oxaloacetic acid. Fumaric acid first form the malic acid and then malic acid to oxaloacetic acid. And during this again NADH2 is produced. So these are the steps which are very important in the in this Krebs cycle, including this conversion of pyruvic acid to acetyl. One step of oxidation here, another step of oxidation is here NADH2 production. Next one is here next is this one and this one. Okay, so we can see that from one molecule of pyruvic acid, what we are getting from one molecule of pyruvic acid, we are getting
We are
what?
getting 4 carbon
1,
and 5.
2, 3, 4 NADH2. We are getting 4 NADH2. Then we are getting one molecule of FADH2, one FADH2, and one molecule of GTP, which is equivalent to one molecule of ATP. So we can write one molecule of ATP, or that is GTP. Okay. So this is what we are getting from one molecule of pyruvic acid. One pyruvic acid. Okay. But you know, this after glycolysis, how many molecule of pyruvic acid we get? We get two molecule of pyruvic acid after glycolysis. So if two molecule of pyruvic acid undergoes this Krebs cycle, then we will be getting, that is, into 2. So it will be equal to 8 NADH2, 2 FADH2, and 2 GTP, or that is equivalent to, it is equivalent to ATP. This is by the oxidation of the pyruvic acid in Krebs cycle. Okay. These are actually, these are NADH2, FADH2. These we call reduced coenzyme. Reduced coenzyme. Okay. This reduced coenzyme, these coenzymes are always required to run the this glycolysis and Krebs cycle. Okay, so these reduced coenzymes are to be oxidized. That is, the NADH2 must be converted into NAD. Okay, this should be converted into NAD. And when this is converted into NAD, there is a release of energy. There is a release of energy, and this energy is utilized for the synthesis of ATP. Okay, for synthesis of ATP. And this process, that the oxidation of reduced coenzyme takes place in a chain of reaction, which is known as electron transport system. right? Electron Transport System, or the ETS. Electron transport system consists of different types of carrier molecule. Here the carrier molecules, they are the carrier of either hydrogen carrier or they are carrier of electron. So mostly electron carriers are there, and those carrier, particularly important carriers, are cytochromes. Several cytochromes are there. Then there is carrier molecules like ubiquinone, ubiquinone. Ubiquinone is mainly it is a hydrogen carrier. Then there is some protein which contain iron sulfur. So we call them FES protein. There is FMN, flavin mononucleotide. Then FAD, also there is FAD, flavin adenine dinucleotide. So these are the carrier compounds. And this electron transport system, that is the placement or the location of the electron carrier, that is the inner membrane of mitochondria. That is, electron transport system is located where? It is located in the inner membrane of mitochondria. You remember that mitochondria has two membrane. One is outer membrane, which is smooth, and inner membrane is folded inward, like forming finger-like structure, the cristae. Okay, they are located in this membrane. In this membrane, there is also one very special structure which is present on the inner face of the inner mitochondrial membrane. Inner face means this is sometimes called M face. M face means matrix phase. This is the mitochondrial matrix. So this towards the matrix it is facing. These are called the fundamental particle or elementary particle. Particle, and best it is named as F0, F1 particle, or F0, F1 ATPase particle. Okay, these are located on the inner surface of the mitochondria. Different different carrier molecules. Okay, different carrier molecule, they are organized as a unit, and this units mm, this unit, there are first unit is there, which we call the complexes. Okay, so there are complex complex 1, complex 2, like this, complex 3, complex 4, and complex 5. Okay, this things you can see here that this from your book you can see okay, this here is, this is the complex 1. In complex 1 it is NADH, complex 1 is NADH dehydrogenase. NADH dehydrogenase. Complex 2 consists of succinate dehydrogenase. Okay, this one complex 2 and it is succinate dehydrogenase. It has FAD and FES protein, iron sulfur protein. Then also outside, of course, so there is ubiquinone, etc., which is outside. Then complex 3. Complex 3, it is also known as cytochrome BC1. Here is cytochrome B, then FES protein and cytochrome C1. This is the cytochrome BC1, cytochrome B, cytochrome C1 is there. Cytochrome C is another carrier which is connecting the complex 3 and complex 4. Complex 4 contains cytochrome C oxidase. Cytochrome C oxidase, actually it is cytochrome A and cytochrome A3. So there are cytochrome A to 4. When electron pass from one carrier to another complex 1 to 4, the electron chain, they are coupled to ATP synthase. ATP synthase that form this one. This one is the ATP synthase. This form the complex 5. So what happened that the hydrogen which is present in the NADH that move from the complex 1 to complex 3 and then to complex 4. This complex 2 is in between complex 2. This complex 2 receive hydrogen from the FADH2. This accept hydrogen from FADH2. And during its passage, the electron moves, the energy is released, and ultimately those energy is utilized for the synthesis of ATP. You can see here. You have to remember this is actually a really complex structure, as name also suggests that. But you have to remember that during the process of electron transport, okay, during the process of electron transport, one thing happened that the hydrogen ions, all right, that is the protons are released in between the two membrane of the mitochondria. That is known as protons released in intermembrane space. Okay, intermembrane space means that that the two space between two mitochondrial membrane. This is intermembrane space. Intermembrane space. All right. This all the protons are accumulated here. So when these protons are accumulated here, there is a formation of proton gradient. This leads to the establishment of proton gradient across membrane, across inner mitochondrial membrane. Membrane. This proton gradient is very, very important for the synthesis of ATP. This is required for ATP synthesis. Synthesis by chemiosmotic process or chemiosmotic principle. Okay, this in general, if we see that the hydrogen from the NADH, it is going to FMN through this iron sulfur protein. It passed to ubiquinone. Ubiquinone release ubiquinone. When the electron come to the ubiquinone, ubiquinone can accept proton from the matrix, and this proton, that is hydrogen, is released on the intermitochondrial space. From here, from the ubiquinone, this electron moves to the cytochrome B, and from cytochrome B it goes to cytochrome C1 through APS protein, and from cytochrome C1 it moves to cytochrome C, and then from cytochrome C it goes to cytochrome A, and then to cytochrome A3. this one. So it is from particularly here cytochrome B to cytochrome C to cytochrome A to cytochrome A3, cytochrome A3. Okay, and this cytochrome A3 pass the electron to oxygen. From here, cytochrome A3 pass electron to oxygen, but it is not done directly. Okay, this is done via copper atom. Okay, this cytochrome A3, say all the cytochrome has iron as a element. Okay, iron elements are there, and due to presence of the iron only, the cytochromes can transfer electron from one cytochrome to another cytochrome, because iron has the reversible valency. Okay, that iron um, plus 3 and iron plus 2. Okay, cytochrome copper also has this thing, copper 2 plus and copper 1. Okay, so by this reversible valency they can accept electron and pass electrons to the other, and so this cytochrome A3, which contain iron as well as copper. So from the iron, electron moves to the copper, and copper pass the electron to oxygen. When the oxygen get two electron, okay, oxygen can accept two electron, and it become negatively charged. Then this oxygen with the negative charge can accept two proton, that is two hydrogen ion, and then it forms water, H2O. This water is formed at the end of the respiration. Okay, so you can see in the glycolysis, in the Krebs cycle, there was no use of oxygen. Oxygen used at the end of the process just to accept electron. Okay, so electron and hydrogen ion, and it forms water. Here is oxygen is used. So, and then there is a process of ATP synthesis, how ATP is synthesized. ATP synthesis, this is known as oxidative phosphorylation. Why it is called oxidative phosphorylation? Because for the synthesis of ATP, it requires energy. And in this process, energy is coming from the oxidation of organic food. So the synthesis or the phosphorylation process that takes place in the respiration, that is known as oxidative phosphorylation. This oxidative phosphorylation, for there is that fundamental particle located in the inner mitochondrial membrane. This fundamental particles, this has two part. Okay, one portion, this is more or less a knob-like structure located in the inner mitochondrial membrane. It has a base. This base portion is F0. This is F0 portion, and to this there is a knob-like part which is exposed outside the membrane. Say this is the membrane and this membrane. So it is base is the F0 portion. These are all protein, okay? F0 portion, and this portion it is F1, okay? And it has this has a enzyme activity. So this is actually ATPase, okay? ATPase, or we can call it ATP synthetase. ATP synthetase. What happens? Say if this one, this is the inner mitochondrial membrane. Say I'll write in short inner mitochondrial membrane. This one here is say outer mitochondrial membrane, and this is the intermembrane space. Intermembrane space. The during the process of this electron transport, what happen in the intermembrane space, there is accumulation of proton. So positive charge is increased in this space, and this side is the matrix of the mitochondria. So here a gradient is this side less proton, and here in the intermitochondrial space more proton. So this proton, they try to move to the matrix, because here it is higher concentration, here it is lower concentration. So they try to move, they try to diffuse in the matrix. But this is not possible, because the inner mitochondrial membrane, it is leak proof, means through this membrane, for this hydrogen ion, this proton cannot move, but they have a passage, and passage is that F0 F1 particle. This one is the passage through which this proton can move. So when this proton moves from the intermitochondrial space to the matrix, there is a release of energy, okay? And when there is a release of energy, this has this F1 particle, F1 portion, they have ATPase activity. So ATP is synthesized from ADP. From ADP, ATP is synthesized. And this hydrogen protons, they are released in the mitochondrial matrix. Okay, this proton, actually uh, sorry, this proton is actually here combined with the oxygen, to form water. And due to this oxygen, that cytochrome oxidase, cytochrome A3 is located on the face, inner face of the inner mitochondrial membrane. For every NADH2, NADH2, there are three ATP molecules are produced, are produced. Okay, because this one NADH2, when it moves in the electron transport system, they can release 6 proton to the inner mitochondrial membrane, either from here, or it help to move the proton from matrix to the intermembrane space. Okay, so there is 3 ATP is formed, but it pass 6 hydrogen atom, 6 proton. So for every ATP, for each ATP, 2 protons are to move from the from intermitochondrial space the uh, to the matrix through this F0 F1 particle, means 2 proton. Okay, then the capacity of the production of ATP for FADH2. FADH2 can produce 2 ATP molecule. Okay, now we have got in the Krebs cycle how many? How many ATP we have got? Say, from the Krebs cycle, including oxidative decarboxylation, we have got 3 NADH2. So each one can produce 3. So 8 will produce as from Krebs cycle. We have got, by the oxidation of 2 molecule of pyruvic acid, that 8 NADH2 So this will produce 3 8 into 3 is equal to 24 ATP. Is it? Each one give 3. We have got 2 molecule of FADH2. So it is producing, each molecule of FADH2 can produce 2 ATP. So there is 4 ATP. Is it? And at the ground level, substrate level, we have got directly 2 GTP. Is it? And this 2 GTP, each GTP can give 1 ATP. So 2 into 1 is equal to 2 ATP. Okay. So here altogether 30 ATP molecules. From the Krebs cycle, from one molecule of glucose in the Krebs cycle, we are getting 30 ATP. We also get 2 ATP in the glycolysis. Net gain in glycolysis is 2 ATP net, 2 ATP, and 2 molecule of NADH2. This 2 molecule of NADH2, remember one thing, this NADH2 which has been produced in the glycolysis, they cannot produce 3 ATP each. They can produce only 2 ATP each. So these 2 will produce into 2 is equal to 4 ATP. So in that case, in the glycolysis, our net equivalent gain is 6 ATP. So from the glycolysis, 6 ATP, and from Krebs cycle, 30 ATP. So total 30 plus 6 is equal to 36 ATP. In some animals, in case of plant, it is the 36 ATP from one molecule of glucose. Okay. In some bacteria, all right, and some of the in animals, sometimes it can produce 38 ATP molecules. Okay, but in general, we know it is 36 ATP from by oxidation of, that is one molecule of one glucose produce 36 ATP molecules. Of course, energy